Hello, good morning, everybody. Today I'm going to be presenting to you the effects of varying dietary enzymes inclusions on the diets of Florida pompanos. Florida pompano is a fish that is found all the way through the West Atlantic coast. It ranges all the way from Massachusetts all the way down to Brazil. The market demand for the Florida pompano is high due to its premium market status and excellent taste. The taste is usually described as mild, or it also has a butter-like texture. And the Florida pompano is a high-value species that has a rapid growth and a high market demand. However, it has a very bad poor feed conversion. This is it was said by Breder in 1926 that this might be because of the swimming involving a subsinural oscillatory movement that restricted the rear body and the tail, while little or no movement of the majority of the body called carangiform swimming or locomotion. Because of this, are characterized as a very fast endurance swimmer. So basically, it is saying it's like you would have a steamboat with a very small furnace, and you're trying to push the fuel in, and you just have such a big powerhouse to push forward, right? So what we decided to do is we decided to try different levels of inclusions of dietary enzymes in our diets. Dietary enzymes like protease and silanase act as biological catalysts by breaking down complex nutrients into more digestible parts, improving the nutrient absorption and reducing the nutritional factors in feed. Enzymes are usually composed of active zone, which the protein comes in, interacts with it, goes through a hydrolysis process, which makes it break down into a final product. In this case, we're going to be having silanase. So we try silane, and we have the final product, which be xylose. And we also have the protease, which break down peptines and amino acids into small peptides. The use of enzymes throughout the aquaculture industry has shown an improvement in weight, fish efficiency, and multiple fish species like now tilapia, Atlantic salmon. However, there has been no change in other species such as the Orchromis micis, which would be the rainbow trout. Our research objectives were to determine the effects of the inclusions of the protease and silanase in Florida pompano diets by evaluating the growth performance and nutrients retention. We also observed the feed efficiency on the different levels of dietary inclusions for protease and silanase. This was a diet that we formulated. We were using mostly plant-based proteins, and we did the different levels of inclusion, being it half the doses recommended by the doses recommended by the company, and twice the doses. We did this with the protease as well as the silanase. To make our experimental diets, we used pre-ground dry ingredients. They were weighed and later on mixed on the mixer. While they were mixing for 15 minutes, we added oil, and we also added boiling water into the mix. Finally, we extruded through a 3 millimeter die to finalize on a forced air drying oven at approximately 45 degrees Celsius, or until it reaches 10%, less than 10% moisture. This was our diet proximate analysis, showing that our diet were formulated to a 40% protein and 8% lipids. Our experimental setup, we set up this experiment on the Claude Petit Mariculture Center in Gulf Shores, Alabama. We used a recirculating aquaculture system composed of 36 tanks with a capacity of 800 liters or zero point 0.8 cubic meters each. Our stocking density was 25 fish per cubic meter, making it a total of 20 fish per tank, with a initial weight of 8.30 grams and a standard deviation of 0.27. Our data was analyzed using a two-way analysis of variance to figure the significant variance between the treatment means. To determine the significant difference between the treatments, we used Tukey's honest significance difference test, and we used the program SAS version 9.4 to run the statistical analysis. To take our water quality and make sure that our fish were having a good environment, we measured twice a day with a YSI Pro Quattro multiparameter, which is this one over here. We measured DO, pH, temperature, and salinities twice a day. We then used a thermoscientific Orion 4-star plus to measure the total ammonia and nitrogen, and we did this once a week. As well, once a week, we also did water link spin touch photometers to measure the hardness, calcium, magnesium, nitrate, and alkalinity. These were our quality parameters throughout the whole investigation, and we measured dissolved oxygen, temperature, salinity, pH, total ammonia, nitrogen, nitrate, nitrogen. This was the final growth performance. This was the response of the Florida Pompano when acclimated to these diets. This is the results. We can see that there were no statistical differences in between the treatments. However, we do see that we have a very a good pattern in between the silanase. We measured final weight gain percentage, survival rate, net protein retention, and feed conversion ratio, and still we didn't find any significant difference between these different treatments. This is our whole body composition. We used the approximate analysis of the Florida Pompano. We measured moisture, crude protein percentage, crude lipids, and ash. We also didn't find any statistical differences in these results. And all of these values are measured in as-is basis. For our conclusion, we saw that there's consistent performance across all the diets. And we also know that silanase protease, or the inclusion of enzymes in fish, is also species-specific response. And then we're still conducting this research, and we're still running digestibility, so we can maybe find something deeper in regards to the energy used in these feeds. I want to thank the aquaculture team on the coast, Club Marine Petite, and the Auburn team at School of Fisheries. Thank you very much.
We have a good time for questions. Any questions? Well, thank you, Tommy. Congratulations.
Thank you.